You enter the water. The ocean looks empty, but something is already there. Something almost impossible to see. It has no bones, no teeth, no claws, no centralized brain. And yet, evolution gave it something far more disturbing: vision, movement, precision, and venom powerful enough to bring the human heart to a stop. You may never see what touched you, but your body will know. In the shallow tropical waters of northern Australia, the threat is defined by what remains unseen. This is Chironex fleckeri, the Australian box jellyfish. Formally recorded in 1956 by researcher Bernard Southcott, this cubozoan possesses a physical design that defies the typical rules of aquatic survival. Its body is almost entirely translucent. The gelatinous tissue of its bell transmits light directly and scatters remarkably little of it underwater, allowing the creature to dissolve into the shifting glare of the coast. An adult bell can reach 12 inches across, roughly the width of a human head. Suspended below are dozens of thin, ribbon-like tentacles that can extend up to 10 feet into the surrounding water. The sheer physical scale of the animal stands in direct contrast to its complete visual absence. In waist-deep water, a swimmer can stand inches away from a massive, multi-tentacled predator and notice nothing but empty space. Yet this raises an immediate, unsettling question about the nature of the unseen organism. Is it merely drifting with the tides, a passive hazard of the shallows? Or is it actively moving, watching, and hunting? The idea that jellyfish are passive drifters does not apply here. Observation reveals a highly capable swimmer. Propulsion comes from rapid contractions of the square bell. At its base lies the velarium, a muscular rim that narrows the opening. By restricting this aperture, the animal concentrates the expelled water into a powerful jet. It can shift this opening to direct thrust, executing sharp turns and altering course with precise control. This capability allows it to hold its position within turbulent coastal waters rather than being swept away by currents. In laboratory settings, kinematic analysis records controlled bursts of speed around 0.4 miles per hour. Some field observations report escape speeds approaching 4.6 miles per hour, though scientists view this higher figure as an estimated maximum burst in the wild, rather than a confirmed laboratory measurement. This contrast in measurement scales explains why daily displacement is measured in yards per hour, while burst capability allows for sudden acceleration. This controlled movement keeps the animal in rich nearshore environments, securing future access to calm feeding grounds. Traversing these shallow zones requires real-time choices. It raises a difficult question. How does an animal with no central brain guide itself through the water? Suspended around the lower margin of the translucent bell are 4 specialized, club-like structures called rhopalia. Each of these sensory hubs functions as a localized control center, containing balance organs, neural tissue, and its own set of eyes. Within these 4 structures lies the major anatomical secret of the box jellyfish: 24. That is the total number of eyes carried by this simple invertebrate. 6 eyes are packed into each rhopalium, divided into 3 unequal designs. 4 of these are simple pigment cup ocelli. Specifically, each club houses 2 pit eyes and 2 slit eyes. These simple structures function essentially as light meters, detecting broad shifts in intensity, basic contrast, and the angle of the sun. But the remaining 2 eyes in each cluster are dramatically different. These are camera-type lens eyes, built with a cornea, a retina, and a cellular lens. The upper and lower lens eyes are physically capable of forming low-resolution images focused on different angles of the marine environment. Rather than a uniform visual system, the jellyfish relies on these non-identical optical tools to gather distinct streams of data. Yet, None of these eyes connect to a centralized brain. The visual system operates entirely within a decentralized network. It forces a fundamental question. How does a creature with no central processing unit integrate these 24 separate visual inputs to make a single, real-time decision? A diffuse nerve net spans the gelatinous tissue, operating without a central command organ. Control instead relies on the 4 rhopalia. Each houses a rhopalial ganglion, a localized processing hub of roughly 1,000 neurons. Adjacent to these neural clusters sit statocysts, balance organs containing calcium sulfate crystals that constantly monitor orientation. When the animal swims, a dedicated motor nerve net coordinates the rhythmic contractions of the bell. This decentralized network performs sensoromotor integration, defined as the direct conversion of sensory input into physical movement. Visual signals do not travel to a brain for analysis. They flow straight to the rhopalial ganglia to drive reflexive obstacle avoidance and course correction. This process operates as a direct reflex loop, entirely separate from conscious thought, planning, or cognitive processing. The mechanics of this distributed control are documented through physical studies. Damaging or removing a single rhopalium immediately disrupts swimming coordination and steering. Fine-grained experiments on related box jellyfish species, specifically Tripodalia cystiphora and Chiropsella bronzi, pinpoint the exact pathways. When researchers selectively cover the lower lens eyes of these creatures, their ability to swerve away from physical obstacles vanishes entirely. The local ganglion simply stops sending the signal to alter the contractions of the bell, proving that movement is managed by these isolated, independent processors. In the shallow reef flats and along the tangled edges of mangrove forests, this controlled movement serves a singular biological purpose. Adult box jellyfish actively seek out small fish, while the smaller juveniles focus their efforts on shrimp, prawns, and other tiny crustaceans. This dietary shift mirrors their physical development. These predators do not simply float and wait for a chance collision. Instead, they traverse the shallow margins with purpose, expanding their hunting range through directed swimming. Their long tentacles trail behind them, forming a wide, highly sensitive net. When the sensory systems detect prey in the coastal shallows, a precise biological sequence begins. It is not conscious planning, but a hardwired sensoromotor reflex. The animal detects, approaches, and makes contact. Once an organism touches the trailing tentacles, the jellyfish uses coordinated contractions of its bell to transport the captured prey upward, drawing it directly toward the mouth-like manubrium. This physical coordination links perception directly to movement, keeping the predator positioned in the richest hunting grounds of the shallows. Yet, this entire hunting sequence relies on a single, instantaneous event. The transition from active swimmer to lethal captor is immediate. What actually happens at the microscopic level, the exact millisecond a tentacle makes contact with its prey? The answer lies hidden inside the tissue of those trailing threads. Along the entire length of the trailing tentacles sit millions of specialized stinging cells known as conidocytes. Inside each cell lies a nematocyst, a highly pressurized capsule containing a tightly coiled, hollow tubule. This microscopic structure functions as a biological weapon. When an object brushes against the tentacle, mechanical contact, sometimes assisted by chemical cues on the prey's skin, trips the biological trigger. What follows is not a deliberate muscle contraction, but an automated physical explosion. In approximately 2 microseconds, the massive internal osmotic pressure ruptures the capsule. The coiled tubule everts, firing outward and turning inside out. It drives forward with immense acceleration, punching directly through tissue layers to deliver the venom stored inside the capsule. This same microscopic apparatus serves both active predation and defense. A fraction of a second of contact instantly transforms the soft, trailing ribbon into a firing platform of millions of microscopic harpoons, breaching the physical barrier of the skin and establishing the direct pathway for the chemical cargo to enter. The microscopic harpoons inject a potent mixture of biologically active proteins. This specific chemical signature belongs to Chironex fleckeri, representing a molecular payload far more immediate in its systemic destruction than other box jellyfish species. At its core are the CFTX1 and CFTX2 toxin families, which belong to a class of molecules known as pore-forming toxins. These proteins disrupt cell membranes by drilling abnormal, unregulated openings through the cellular walls. Alongside these destructive pores, the mixture contains metalloproteases, which break down tissue structure, and phospholipase A2, an enzyme that drives rapid inflammation and localized cell damage. The physiological response to this cellular assault unfolds along a rapid clinical spectrum. Contact instantly triggers excruciating, blinding pain, followed by severe skin lesions and deep tissue destruction along the exact path of the tentacle contact. In serious envenomations, this localized trauma quickly escalates to systemic devastation, causing acute respiratory distress and severe cardiovascular disturbance. In the most extreme cases, where a large volume of venom enters the bloodstream, the heart fails, progressing to cardiovascular collapse, cardiac arrest, and death in under 5 minutes. Yet, these life-threatening outcomes are not a guaranteed sequence. The clinical severity of a sting is highly variable, dictated primarily by the total length of tentacle contact and the speed of medical treatment. Many encounters result only in painful local injuries and scarring, demonstrating that the venom's ultimate impact depends entirely on the physical dose delivered to the body, rather than an inevitable fatal pathway. When a high dose of venom enters the bloodstream, the focus of the emergency shifts from the skin to the cardiovascular system. The superficial scarring and local tissue damage fade in comparison to a deeper, quiet cellular threat. The primary target is the heart itself, specifically the cardiac myocytes, the specialized muscle cells responsible for keeping the heart beating. The CFTX proteins in the venom initiate a destructive pore-forming process. They insert themselves directly into the membranes of these cardiac cells, drilling tiny, unregulated openings through their protective walls. This physical destruction leads to an immediate, catastrophic shift. Ions flood across the damaged membranes in an uncontrolled wave, short-circuiting the heart's delicate electrical pathways and disrupting its mechanical function. The heart muscle loses its ability to contract normally. As contractility drops, the heart struggles to pump blood, initiating an unstable, erratic rhythm. Arrhythmia takes hold. This rapid cellular breakdown is the exact mechanism behind the systemic emergency. It is why severe envenomations demand immediate hospital evaluation. At this stage, the localized skin injury becomes secondary to the rapid mechanical failure of the circulatory system. This threat concentrates at the edge of the sea. In northern Australia, public safety warnings focus heavily on the traditional stinger season, running from October 1st to June 1st. Yet, this window serves as a practical risk management tool rather than a strict biological boundary. The animals do not follow human calendars, and encounters occur outside these months. The primary hazard exists in the shallows. Surveillance records from the Northern Territory show a clear pattern. Approximately 83% of confirmed Chironex fleckeri stings occur in water 3.3 feet deep or less. While these emergency records do not represent a complete census of the jellyfish population, they map where human activity intersects with the predator. This ultra-shallow zone is where people swim, wade, and walk along the shoreline. It is also the prey-rich coastal habitat where the jellyfish actively hunts. The danger is a near-shore reality. This spatial overlap raises a broader scientific problem. When warning signs blanket the coast, they often use the generic label box jellyfish. But with roughly 50 described species in this class, ranging from massive predators to tiny single-tentacle organisms, this broad term obscures the specific dangers posed by Chironex fleckeri. These 50 species vary wildly in physical scale, habitat, and the clinical impact of their venom. While Chironex fleckeri represents the giant of the class, other species are nearly invisible to the naked eye, yet carry their own distinct, systemic dangers. Among these smaller relatives are the Charybdid jellyfish, most notably Carukia barnesi. These tiny organisms, with bells measuring less than an inch across, are responsible for Irukandji syndrome, a medical emergency entirely separate from the rapid destruction caused by Chironex fleckeri. An Irukandji sting is often so mild, it passes unnoticed, causing no immediate local pain or rapid cardiac failure. Instead, the danger is delayed. Between 30 minutes and 2 hours after contact, a systemic catecholamine surge floods the human body. Victims experience severe, generalized muscle cramping, soaring blood pressure, and a racing heart rate. By grouping these vastly different organisms under a single fearsome name, the true complexity of their survival strategies is lost. Each species has carved out its own precise ecological niche, leading to distinct evolutionary solutions. At the heart of this animal's survival lies a series of stark physical contradictions. An organism entirely devoid of a centralized brain manages 24 eyes and translates visual inputs into highly coordinated sensory motor behavior. Its fragile, almost completely transparent body, which seems built to drift helplessly, instead executes active, directed swimming through turbulent, wave-swept shallow waters. Lacking jaws, teeth, or claws, this simple invertebrate achieves highly effective prey capture using a chemical weapon of unparalleled potency. The long, trailing tentacles, visible as they sweep through the water, achieve their decisive action at a microscopic scale. Millions of pressurized stinging cells fire in microseconds, launching a cellular assault that can trigger catastrophic, rapid cardiovascular collapse in a victim's body. This design demonstrates that evolution does not need to reproduce the human arrangement of a centralized brain, paired eyes, integrated muscles, and cognitive awareness to achieve complex survival tasks. Instead, the box jellyfish relies on a highly efficient modular solution. The 4 Rhopalia, working in tandem with the distributed nerve networks, act as self-contained sensory motor loops. For this creature, seeing does not mean experiencing the surrounding world in the rich, conscious way a human does. It is defined simply as the extraction of usable information from light, such as detecting a shadow or sensing the tree line to drive immediate physical action. Its coordinated behavior is a series of precise reflex loops, entirely distinct from human-like intelligence, memory, or intent. Yet, this decentralized setup leaves scientists with a profound, unresolved biological mystery. Researchers still struggle to explain how these separate, localized nervous systems coordinate their individual inputs to produce a single, unified behavioral response across the entire organism. If each Rhopalium processes its own visual data independently, how does the animal decide to turn left rather than right when multiple obstacles appear? This puzzle reframes how we view Chironex fleckeri. It is not an evolutionary dead end, a biological mistake, or a mindless ocean monster. It is a highly successful, radically different evolutionary solution to the universal challenges of vision, movement, hunting, and self-defense. The strangest thing about the box jellyfish isn't that it can kill. It's that something with no centralized brain can see, swim, navigate, and hunt. Nothing about it is normal. As science maps the limits of animal consciousness, this phantom of the shallows forces us to redefine perception itself. The strangest thing about the box jellyfish isn't that it can kill. It's that something with no centralized brain can see, swim, navigate, and hunt. Nothing about it is normal.